So next, we're going to hear from our first of this year's new group of NGLs. We're going to be hearing from Mark Cembrowski. Mark is currently a postdoc at the Spatial Laboratory at the Genelia Research Campus, and his postdoctoral work combines experimental and computational approaches to examine the extent of heterogeneity within classical cell types of the hippocampus. In, 3 weeks, Mark is going to be starting his own lab up at the University of British Columbia at the Center for Brain Health. His lab's going to examine how molecular and cellular properties of the brain create, maintain, and transform memories. Mark received his PhD in applied mathematics from Northwestern University as a joint student between William Kath, Herman Reike, and Joshua Singer. His talk today is titled Cell Type Specific Rules of Hippocampal-Dependent Memory. Please join me in welcoming Mark.
All right, good morning, everyone. It's really an honor to be here today to present my research. My name again is Mark Cembrowski. I'm a postdoc in Nelson Spruceton's laboratory at Genelia, and in a few weeks I'll be starting up my own laboratory at the University of British Columbia. In Nelson's lab, as well as in my forthcoming lab, we study the hippocampus in the mouse. And the hippocampus is an absolutely fascinating brain region to study. I realize right off the bat that I'm a little bit biased, but ultimately I think there are some really compelling features to this brain region. And I'll highlight just a few here. So well understood is the role that the hippocampus plays in things like learning and memory, spatial navigation, and in emotion. And more recent experimental evidence has come forward that suggests the hippocampus can be involved in things like encoding representations of the passage of time, features of reward and motivated behavior, as well as encoding other features of the external environment like auditory frequency. So a natural question to ask is, how does this single structure give rise to so many different hippocampal-dependent behaviors and functions? This question is especially interesting when we consider the circuitry of the hippocampus shown here. Within the classical trisynaptic loop of the hippocampus, input arrives at dentate gyrus granule cells. These granule cells signal the CA3 pyramidal cells. CA3 in turn signals the CA1 pyramidal cells, and within the context of this trisynaptic loop, these CA1 cells are the output cells. For today's talk, I really want to emphasize that in the dorsal hippocampus, which will be the focus of my talk today, CA1 talks primarily to its next door neighbor here, the subiculum. And it's the subiculum that can be viewed as the primary output region of the hippocampus, both in terms of the total number of downstream structures, as well as the density of projections to any one of these structures. So how do we rectify this relatively straightforward structure with the relatively complex function of the hippocampus? Well, one answer might lie in that each one of these classical cell types that I just described here might actually be a whole host of heterogeneous cell types. And this dissociable function might therefore produce dissociable structure, might produce dissociable function and computation. This is not a new idea, and indeed some of the pioneering work that's been done on this was here at the Allen Institute with Carol Thompson and others. My research today will focus on this heterogeneity specifically from the perspective of memory, trying to understand hippocampal-dependent memory across these specific cell types and across the stages of memory, like the formation of a memory to the subsequent retrieval. I've been working on this question over the last few years as a postdoc, examining all of these classical cell types that I previously mentioned. And what we found is that there's heterogeneity within all of these classical cell types. My goals have been threefold: to understand to what extent there's heterogeneity present, to come up with the spatial organization underlying this heterogeneity, and then finally to map it onto functional properties like memory. My brief talk today will focus on two papers here that came out earlier this year. And both of these papers focus on the subiculum, and in particular pyramidal cells in this region. These are the projection cells that talk to extrahippocampal regions. At the heart of my talk today is a very straightforward question. Are all subicular pyramidal cells performing the same computation and giving rise to the same hippocampal output? Naturally, there's been a lot of work that's been done in this regard already. I'm going to highlight one conceptual model here. This is a model that's been put forth by Jim Knierim. And for the moment, I'm just going to abstract this model to its salient details. Jim has proposed, based upon a combination of anatomy as well as in vivo recordings, that there can be two streams of information that flow through the hippocampus. One stream of information is involved in representing elements of the local environment. The other stream of information is involved in representing elements of the distal or more global environment. Within the context of this conceptual model, different subregions of the subiculum are differentially involved in processing this local and remote environment. Now, naturally, this conceptual model that I just discussed here is rather high-level behavioral systems. My goal for today's talk is to try and flip this. Start at the opposite end of the spatial scales of the nervous system, identify heterogeneity in the molecular properties of the subiculum. Use this to establish a framework with which we can then understand heterogeneity at higher-order cellular, circuit, and behavioral level properties, hopefully to recapitulate this conceptual model, but also to lend underlying mechanistic insight. I'll give away the punchline of my talk today. It's that this classical region, the subiculum, can actually be subdivided into two discrete abutting subregions. I'm going to call these subregions proximal and distal today. Geographically, proximal just means close to CA1, distal means farther away. Don't worry about specifically where these are located. All I want you to take from this is that there are two subregions that sit right next to each other, and they can be coherently divided across molecular, cellular, circuit, and behavioral properties. For the ease of you, the listener today, I want to emphasize that everything that deals with proximal subiculum will be colored green. Everything that deals with distal subiculum will be this purple magenta color. Also, for the aficionados in the crowd, I want to emphasize that there is a rough equivalency of proximal and distal subiculum to pro and proper subiculum, but there are also some differences which I'm happy to discuss offline. So let's start at the molecular level of the subiculum. As I mentioned before, subicular pyramidal neurons project to a whole host of downstream targets. These targets are shown here. Don't worry about the details. All I want you to take from this slide is that there are a lot of downstream targets. And the conventional thinking is that one subicular pyramidal cell projects to exactly one of these targets. So I have to recouch my initial question here. Is a pyramidal cell that projects to region X and another pyramidal cell that projects to region Y otherwise interchangeable? We can begin to address this question from the perspective of next-generation RNA sequencing. Here in a given animal, we would inject two downstream targets of the hippocampus with different colored retrograde beads. For example, the nucleus accumbens, shown here, and the retrosplenial cortex, shown here. These beads are retrograde. They're picked up by the axons. They backlabel cell bodies in the subiculum. At left here is a coronal section of the hippocampus, which includes the dorsal subiculum, our region of interest, shown in yellow. This is a zoomed out version of this bead labeling, and if we expand, we see a very striking spatial profile emerge. Cells that project to the nucleus accumbens, which are colored green, are located proximally but not distally. Cells that project to the retrosplenial cortex, which are colored magenta, are distal but not proximal. And there's a rather sharp dividing line that separates these two projection classes. Next, we harvested these cells for next-generation RNA sequencing, so we microdissect out this region, dissociate this tissue, and then manually purify for two pools of colored cells, pure green cells and pure magenta cells. For today's brief talk, I'm just going to discuss population-level RNA-seq, but I want to emphasize that we've also done some single-cell RNA-seq that recapitulates and extends what I'll show you here right now. So when we look at the transcriptomes associated with these two projection classes here, an easy way to visualize this is a scatter plot, where on the x-axis is how strongly each gene in the genome is expressed for cells that project to the retrosplenial cortex. On the y-axis is how strongly each gene is expressed for cells that go to the nucleus accumbens. If these projection classes are equivalent in their gene expression, all of these points should lie along the line y is equal to x, up to the biological and technical noise. But there's a lot of points that fall off of this line. And if we do differential expression analyses based on, say, a 5% false discovery rate, we see that there are 452 of these differentially expressed genes. This is a big number. This is on the order of what we see when we traditionally compare very different cell types, like granule cells to pyramidal cells. So to see this degree of heterogeneity within a classical cell type of the hippocampus is rather striking. I want to emphasize as well that this degree of heterogeneity isn't just numerically large, but it covers every functional category that any of us as a neuroscientist in the room today care about. Categories from axon guidance and adhesion to peptides and receptors to synapses to voltage-gated channels, suggesting that there's probably a lot of higher order differences between these cells as well. As I mentioned before, we are able to recapitulate these results looking at other projections, as well as with single-cell RNA-seq, suggesting that it's this proximal distal difference rather than projection per se that explains this variability in gene expression. We're also able to map this spatially. For example, we did this in a few different ways. One was using single color in situ hybridization from the Allen Mouse Brain Atlas. That's been a fantastic resource for this study and other studies, and I thank you very much for that. We also did our own two-color fluorescent in situ hybridization, which gives us marker genes that nicely delineate almost all the cells that are proximal subiculum or almost all the cells that are in the distal subiculum. So what we've shown so far is that there's a wide degree of heterogeneity in the molecular properties of subicular pyramidal cells. Can we now map this on to cellular properties? One feature of heterogeneity that's been known for quite some time in subicular pyramidal cells at the cellular level is their action potential trains. If one cuts brain slices and performs whole cell patch clamp recording from cells and drives these cells to fire, you can see one of two firing phenotypes. Some cells exhibit regular spiking, other cells exhibit bursting. Work from others, including Cembrowski, has shown that there is a proximal distal bias to where you find these regular spiking and bursting cells. So a natural question is, do these align with this proximal distal divide that we found before? Of course, we need a way to just visualize this line. So to do this, in a given animal, we inject the nucleus accumbens with red retrograde beads. We cut brain slices, and then we record from cells across the proximal distal axis. Here's an example of one slice where we've recorded from 4 cells, which are shown here in teal. We can record their action potential outputs and then figure out how that action potential output changes as a function of where they sit in the proximal distal axis. As has been shown before, some of these cells were regular spiking cells, some of these were bursting. However, in recording 40 to 50 cells, we found that there was a striking spatial profile associated with these action potentials. For all cells that were proximal up until about a narrow transition region, all cells were 100% regular spiking. There was a narrow, about 200 micron stretch where we saw about a 50-50 split of regular spiking and bursting cells. This is the same area with which we saw some mosaicism of our marker gene expression. And then beyond this divide, all cells that we recorded from were 100% bursting. So the spatial organization that we identified at the transcriptomic level maps on to the heterogeneity we see at the cellular level. Next natural question is, can we now relate this to circuit level properties as well? I gave away a little bit of this at the beginning of my talk. I mentioned that proximal and distal subiculum differentially project to the nucleus accumbens and the retrosplenial cortex. Is this the biological exception or is this the biological rule? To address this more comprehensively in a series of pairwise retrograde injections, I injected a host of downstream regions. And what we found is that for the vast majority of downstream targets of the subiculum, proximal and distal subiculum had entirely different suites of projections. Proximal subiculum had strong projections to the nucleus accumbens, prefrontal cortex, and lateral entorhinal cortex. Distal subiculum instead showed projections to the retrosplenial cortex, the ventral hypothalamic nucleus, and the medial entorhinal cortex, giving us retrograde information that suggests proximal and distal subiculum have largely different downstream targets. This, of course, is retrograde evidence. It would be nice to pair this with anterograde evidence, and so in collaboration with the mouse-light team at Genelia, we did exactly this. We filled individual cells and traced their axons across the full extent of the mouse brain. In this brain, I'll illustrate 3 cells in particular. One cell is green. This is a putative proximal cell. One cell is red. This is a cell that projects to the thalamus. This is actually one of the few exceptions we found before. And then the final cell is our magenta cell. You notice that our green cell, our putative proximal cell, sends an axon anteriorly. Sends a strong ramification to this region here. This is the nucleus accumbens in the green shell, and also has projections that course ventrally into the hypothalamus. This red cell that I emphasized here, this is a special and sparse rare cell that projects to exactly one location, which is one nucleus in the thalamus, and has a strong ramification there. And then in purple is our putative distal cell. This cell does not have the same anterior projections that our other cells has, but rather had projections that would move posteriorly. It would innervate the retrosplenial cortex, which is this magenta cell here, as well as areas of the entorhinal cortex. So this provides direct single-cell evidence that there are different suites of targets associated with proximal and distal subiculum. Now, finally, can we identify behavioral correlates for proximal and distal subiculum? To answer this question, we, of course, need a hypothesis to begin with. One of the neat features that emerged from our circuit level mapping is that proximal and distal subiculum differentially innervated the lateral and medial entorhinal cortex. This might not sound like a big deal. Lateral and medial entorhinal cortex have the same middle and last names. But indeed, they actually have very different representations of the external world. So if one records from cells in the entorhinal cortex as an animal explores the spatial environment, you see firing locked to local objects within this animal's environment. If you move these objects, the firing field can actually follow that object. If you remove the object, that firing field can disappear. This sort of firing lock to local objects is not seen in the medial entorhinal cortex. Rather, in the MEC, this is where you begin to see grid cells, head direction cells, boundary cells, receptive field properties that are locked to more global features of the animal's environment. And in fact, it's these receptive field properties that form the backbone of this conceptual model that I discussed before. On my previous slide, I abstracted this to a few details. Here, I'm going to keep the entire cascade up. In red is the transmission of local information, and in teal is the transmission of distal or global information. And within this conceptual model put forth by Jim Knierim, he suggested that the proximal and distal subiculum might differentially relay these two types of information. So what we might have uncovered from our structural analyses are the cell type specific rules that could underpin this cascade. I'll emphasize here that within this conceptual model, proximal subiculum is believed to represent the nearby environment. Distal subiculum is putatively involved in representing the far environment, or said a little bit more colloquially, proximal to proximal, distal to distal. So how do we assay this in a behavioral experiment? Well, one convenient experiment that we can do is the Morris water maze. If you're not familiar with this task, this involves taking an animal and placing it into an opaque pool of water. Somewhere just beneath the surface of that water lies a hidden platform. The animal would much rather be on the platform than to swim, so it's naturally motivated to find the platform. And it remembers where this platform is based upon extra maze cues that are present in the room. Importantly, there's no local information that the animal has to use to solve this task. Thus, within the context of this conceptual model, this should depend entirely on distal subiculum and not proximal subiculum. To investigate this, we next use our single-cell RNA-seq or our RNA-seq data to identify marker genes that gave us selective access either to proximal or distal subiculum in two different Cre lines. And then we use Cre-dependent viruses, Brian Ross HM4D, inhibitory dreads, and targeted these receptors to either proximal or distal subiculum in a Cre-dependent way. On its own, this receptor just sits dormant. However, when it meets its corresponding ligand, CNO, this induces silencing in the subiculum in our targeted cell types for 1 to 2 hours, which we validated through anesthetized recordings. And importantly, CNO does not need to be administered locally. It can be administered intraperitoneally, circulates, and crosses the blood-brain barrier to produce this effect. To test the animals in the Morris water maze, each animal would receive 4 trials a day. This is an overhead view of the maze, and this small circle is the hidden platform. These X's denote the start locations on subsequent trials. They receive 4 trials a day and 9 days of training in total. Importantly, this platform was moved to a new location each day. This means that the task conforms to a spatial working memory variant of the task. That is, the location of the platform on the previous day does not help the animal to find the platform on the current day. This will become very important later. After these animals get trained for 9 days, we then have 2 test days. On each one of these test days, we administer an agent 15 minutes before the experiment, before the animals are even moved into our experiment room. On day 1, a given animal receives either CNO or vehicle. The animal goes through 4 trials, as in any other standard day. But then after this, there's a 5th probe or free swim trial, where I physically remove this platform from the maze, and we just observe the animal's trajectory over a 1-minute swim. On the 2nd day, we do exactly the same thing, except we flip the agent that we administered. This allows us to have a within-animal control that allows us to compare the silenced case with the control case. In these next graphs, it says 1-minute free swim periods that we're going to compare or visualize and analyze. So an example trajectory is shown here for an animal where this inhibitory dread is targeted to proximal subiculum, but we just administer vehicle. You can see here the trajectory of the animal preferentially spends time swimming in around where the goal is. This can be seen, for example, by comparing to the opposite side of the maze. In the presence of CNO, this animal can still recall exactly where this platform sits. In a different animal, when we target the distal subiculum in control conditions, again, this animal preferentially swims in around the goal. However, when we silence distal subiculum in this animal, this is where we saw a striking phenotype. This animal looked like a naive swimmer and spent no more time in around the goal than any other location. A way of quantifying this is to split our maze into 4 parts and simply quantify the amount of time that the animals spend in the goal quadrant. These, of course, are just representative data from single animals. This is our group data. What we see is that there's a striking drop in performance when these inhibitory dreads are targeted to distal subiculum and we silence this region. Silencing proximal subiculum has no effect on this task, totally consistent with our conceptual model that we proposed before. Now, why does this emerge? First, we looked for trivial off-target effects that could produce these deficits. We saw no deficits associated with anxiety, with locomotion, with novelty. But I think the most compelling negative control that I can show you here today is a reference memory version of the task. In this task, in different animals, we would train a given animal for 5 days with the platform being in the exact same location. After these 5 days, we have these 2 test days as before. And here we see no deficit in behavior whatsoever. This task does not depend on the distal subiculum, suggesting that's a spatial working memory variant of the task that requires the distal subiculum. Now, as I mentioned before, a memory deficit can arise from sort of the 2 sides of the memory coin. It might be a deficit in the formation or the encoding of a memory. It might be a deficit in the subsequent retrieval. Can we identify which stage of memory is associated with this deficit? Or potentially both, as these aren't necessarily mutually exclusive. To answer this question, we leverage an interesting phenotype that our animals have. If we train an animal for 4 trials on a given day with a given platform location, on the next day, we can observe their behavior on the first trial. They preferentially swim near the previous day's goal trial. This allows us to then look at how this first trial swim occurs in the presence of CNO versus on the day after CNO. In particular, if we see a deficit on the day where the animals receive CNO, this points to a retrieval impairment, in that the platform location from the previous day can't be recalled. Alternatively, if you see a deficit on the day after we administer CNO, this is an encoding impairment. The animal did not encode where the new location of the platform was. When we searched for evidence of a retrieval deficit, we didn't find it. Animals could recall the previous day's platform just fine on the day where they received CNO. However, on the day after they received CNO, this is where we saw a precipitous drop in performance, and the animals performed no better than chance levels. This suggests that the distal subiculum was required for the encoding, but not the retrieval of the spatial working memory task. I think this tells us some really interesting things about the hippocampus. I'll briefly cover these. First and foremost, as I introduced it, the hippocampus is classically thought of as a serial circuit. However, what I've illustrated here is that the primary output region of the hippocampus seems to be dissociable, multimodally both in structure as well as in function. This motivates us now in further work to try and go backwards throughout the circuit and try and understand how this dissociable structure and function can be supported by CA1, CA3, and DG. The other interesting feature is that this points to the subiculum being a short-term memory storage device for this particular task. And I also think this is a powerful illustration of how cell types can tell us a lot about the function of the nervous system. We eventually got to this function by leveraging circuit properties to identify molecular heterogeneity. This allowed us to identify an underlying spatial organization of this heterogeneity that we mapped onto cellular and circuit properties. And then we were able to flip this to interrogate behavior. We used our circuit level properties to generate a hypothesis. We used our molecular characterization to get marker genes, and this allowed us to do that cell type specific silencing that produced our deficit. Ultimately, this helps us get to the goal that I stated at the beginning of my talk, trying to understand memory across specific cell types and stages of memory by taking higher order behavioral and systems levels models and being able to deconstruct them according to underlying mechanistic principles. Now, very briefly, as I mentioned before, in addition to that population-level RNA-seq data that I showed you, we've also done an abundance of single-cell RNA-seq. In a more recent eLife paper, we published the transcriptomes of about 2,000 subicular cells. We identified 8 classes of subicular cells in total. There's a whole host of these that are present in the ventral subiculum, which we did not probe in our cell paper. And we also did recapitulate this proximal distal divide, which I discussed at length today. And then we were able to cross-validate these results using in situ hybridization, immunohistochemistry, and then align these various cell types with specific circuits. To help bring these data to the broader scientific community, we've uploaded them to hipposeq.genelia.org. These, in addition to our data going up on this website, we've also augmented it with easy-to-use analysis and visualization tools with the hope that this helps the community to do their own analyses to help our data further their cause. And importantly, it's not just this data set that's been uploaded, but data from all of our previous studies have also gone on this data set. And we hope to have a very comprehensive understanding of hippocampal transcriptomics through this website. Very briefly, I'll touch upon the goal for my laboratory in the next few years, and this is to generate a bottom-up mechanistic understanding of fear memory. In this previous work I showed you, behavior was tacked on at the end. What I want to do going forward is start with the behavior and be able to deconstruct the underlying molecules and cells. Fear memory in this way is rather compelling. Fear memory and fear itself generally keep us alive, so they're ethologically relevant. Experimentally, this is a very tractable paradigm, and there's some potential therapeutic viability associated with this as well. One of the ways with which we'll assay fear memory is in contextual fear conditioning. This is single trial learning, which is very important for our purposes. This involves taking an animal and placing it into a novel spatial environment that the animal has not experienced before, allowing the animal to have a few minutes there, and applying a couple of very mild foot shocks. This produces this aversive associative memory. And this can be externally read out by taking the same animal and reintroducing it to the spatial location, say a day or a week later. You can observe this fear memory by watching the animal exhibit behavioral freezing. That is the absence of movement aside from respiration. This is your external readout of the animal's internal space. One of the strategies that's used in the community to identify cells that are involved in the encoding or the retrieval of these fear memories is to have ways of tagging cells in both of these stages. For example, you can use transgenic animals that selectively tag cells that are involved in the encoding phase. And then you can use immunohistochemistry, say for immediate early genes, to identify cells that are active during the retrieval phase. And then compare cells that are encoding only, retrieval only, or participate in both stages of memory. Our approach is instead of having a histological preparation, we have a single-cell RNA-seq-based readout. In this assay, we play the same game, but at the end of the day, we sequence these cells here. Embedded within the transcriptome, of course, we've got information on the cells that are involved in the encoding. We can identify also the cells that are involved in retrieval. But importantly, we can also relate these participant cells back to the subtypes that we've identified in previous studies, allowing us to get at the cell type specific logic of maybe subtypes that participate in this sphere memory, or alternatively, maybe there are other subtypes that are excluded from participating in this sphere memory. Additionally, because we're doing transcriptomics here, we can also look across all of the transcripts that are present in these cells and identify genes that are up or down regulated, correlated with fear memory, and then complement these with interventional experiments using things like CRISPR-Cas to get at causal molecules that are important for fear memory. It's our ultimate hope that we can use an approach like this to get at the specific cellular and molecular rules of fear memory. We hope that this provides us a framework for comparing and contrasting other forms of memory. And in identifying molecular substrates for fear memory, hopefully this paves the way for interventions for fear memory disorders like those that occur in PTSD. So with that, I'm happy to conclude. All of these people that are here on this slide today made fantastic contributions to the research I presented. And an especially big thanks to Nelson Spruceton, my PI, who's been absolutely supportive. And finally, thanks to you and your time. It's an honor to be here today. Cheers.
Thank you, Mark. That was wonderful. All right, we have a few minutes for questions. So again, if you have a question, raise your hand, and we've got microphones that will come to you.
In one of your slides, you had a 3D model of the brain where you had 3 reconstructions of the cells and showing where they projected to. I was just wondering how you got that video.
So we had that was done in collaboration with the MouseLight team at Janelia. So they have a great platform that supports exactly those analyses here.
Thank you.
Tim.
Hi, Mark. I really enjoyed your talk. Can you speculate on the importance of bursting to the encoding of the FAR?
This is a great question. I could make some very speculative guesses. We know generally that bursting is more efficacious for driving vesicular release and things like that. So maybe there's some safety that's present in the bursting cells that's not there for regular spiking cells. We do know, so all of this classification is done ex vivo in slice work. When you do the same analyses in vivo, you do see general excitability going up, and cells that are regular spiking in this characterization can exhibit bursting. Their cell type phenotype remains the same in vivo, but the excitability goes up. So I'm a little bit reluctant to speculate in a functional setting at this point.
Do we know what specifies the identities of anterior, posterior, like these 2 different parts of the subiculum, and what makes a cell fall into this weird, rare class that's in between?
Yeah, this is one of the questions we're actively pursuing here. We don't have any good information on this. We would love to get an answer for this, and would love any thoughts that anyone here in the building would have on this too.
Any other questions? All right, why don't we thank Mark again for that talk.